this immunology seminar series. So I'm going to be talking
just to interrupt,
yeah.
You can either address questions by adding to the chat, and either I'll read them or ask you to read them at the end, or if there's something pressing, you know, just interrupt Kai the way we do the seminar.
Absolutely. Interrupt me at any time if there's any questions coming up, and I'm trying to pay attention to the chat function. Eric, maybe you can keep an eye on that too. If there's
I'll keep an eye on it, see if anything comes up.
All right, sounds good.
All righty.
So great introduction. Thanks, Eric, for that. I don't need to talk much about my background. So I'm going to be talking today about a project in the lab that's focusing on defining immunosuppressive myeloid cells in the context of breast cancer. And we're trying to go after these cells in single cell and whole organism resolution. So today, May 28, 2020, or according to you know, the current calendar, day 70 of COVID-19 lockdown in California, at least if my counting is correct. So yet another Zoom meeting. Maybe we can move over to a different format soon again, but for the time being, I think this works pretty good. So just to give you a little bit of background about what my lab is interested in. So we're very interested in everything microenvironment. So obviously, when I say microenvironment, I'm talking about the cellular microenvironment, and in the sense that no cell is an island, meaning that no cell is just an isolated entity out there in empty space. So this thing here in the middle is supposed to be a cell, not an eyeball staring back at you. But as you know, cells in complex tissues are embedded into a three-dimensional microenvironment consisting of neighboring cells that oftentimes form close cell-cell interaction with our cell of interest. And then cells are embedded into a three-dimensional extracellular matrix, which surrounds all the cells and provides a lot of the tissue integrity. And then, of course, there are secreted factors and ligands that act on cell surface receptors and really change the behavior of our cell of interest. And all these sort of extrinsic cues have to be integrated together with our intrinsic wiring of the cell. And obviously, considering how complex the human body is with all these different niches in the various tissues of the human body, this is obviously a great source of cellular heterogeneity, which is another aspect of biology that my lab has become very obsessed with over the recent years. And so the system that we're studying is the breast epithelial system, for the reason that this is where breast cancer arises. So we want to learn about how breast cancer develops and how it progresses and in later stages of breast cancer. So breast cancer comes from the breast epithelial system, which is sort of, I'm not going to talk about the details here, but just briefly, the breast epithelial system is a ductal epithelial network embedded into an adipose-rich tissue of the breast. And you have various different regions here, for example, these terminal ductal lobular units, more inward located in the breast. And here you can see sort of the tissue histology view on that. So this like really densely packed epithelium. And within these lobules, after pregnancy, really this is where the milk production takes place. And then you have an intricate system of ducts that you know, has the function of really channeling the milk towards the nipple so that we can feed our offspring after birth. Cross-section here through this breast epithelium. Like I said, I'm not going to get into the details of this, although this is really also something that my lab is very interested in, the distinct cell types within the breast epithelium and how they are maintained in the context of normal tissue homeostasis. And then really, when breast cancer occurs, this, like I said, happens within the breast epithelium. Some of these cells gather oncogenic mutations and they start to proliferate in an uncontrolled manner, which then really starts to change this local microenvironment quite dramatically. So going from normal homeostasis to early tumorigenesis. And then in these later stages of tumorigenesis, we get fairly massive influx of immune cells, inflammatory cells, and the local tissue microenvironment, the local stromal niche is dramatically changed due to the growing cancer. and the different types of signals that are floating around within this tumor microenvironment. And then, as you know, in later stages of cancer progression, some of these cells are able to disseminate out and set up shop at different sites of the human body and really set up metastases, for example, in the lung, which in the case of breast cancer is really the main cause of lethality. So in this grand scheme of breast cancer progression, we're really very interested in all aspects here. So we have projects in the lab that are focusing on normal homeostasis and mapping out the distinct cell types within the normal human breast, and you know, what composes the stem cell niche and maintains normal homeostasis in the breast and in the mouse mammary gland. And then really this arrow here is very interesting to us. How does the tissue change during cancer initiation or even precancerous changes, such as in BRCA1 mutation carriers? I'm not going to talk about these first two areas here. So today's project is really focusing on later stages of tumor progression. When we have a breast cancer present, there are cells within the tumor microenvironment that can very actively suppress an anti-tumor immune response. And this is obviously very bad for tumor progression. And so we want to learn more about what are some of these components within the tumor microenvironment that can suppress an active anti-tumor immune response. The ultimate goal here is to possibly be able to modulate these aspects of the microenvironment in order to improve cancer immunotherapy. Without going into too much detail here of what cancer immunotherapy is, I think most of you are very familiar with this idea. But just in a nutshell, obviously in a normal situation, the tumor cells are being recognized by adaptive immune cells, by T lymphocytes, and eradicated fairly efficiently. At least that's the hope. But what oftentimes happens is that this tumor cells can undergo this camouflage, and they sort of express these PD-L1 molecules, which then are able to sort of signal to the surrounding immune cells and tell them, hey guys, everything is okay. You don't need to kill the tumor cells, and these immune cells then become sort of exhausted, and obviously this is a target that we're trying to get at with these checkpoint inhibitor classes of drugs, such as PD-1 and CTLA-4, and then in order to really raise and unleash this immune response against the cancer. But the problem sometimes can be that the tumor microenvironment is very heterogeneous, and there are numerous cell types infiltrating the tumor, all listed down here in this nice illustration. And what happens, some of these cells can actually interfere actively with this anti-tumor immune response. One of these cells being neutrophil granulocytes, which you can see here, which with their very characteristic polymorphic nucleus here. So neutrophils are essentially one of the first lines of defense of the innate immune response. You know, they come from the bone marrow, where they develop through granulopoiesis, and they're massively produced every day. And in the human body, they're actually one of the most abundant circulating immune cell types. In fact, they are the most abundant peripheral blood leukocyte in the human body, and they survey almost all parts of the human body, including the lung, which is a common site of breast cancer metastasis. They're very prominent in the spleen as well, which are some of the organs that we're particularly focused on in the study today. Just a bit of a little trip down memory lane for me. I actually used to work a little bit on neutrophils myself during my PhD work, and I was particularly interested in a specific class of neutrophil serine proteases and how they are able to regulate an inflammatory response by changing aspects of the local tissue microenvironment here in the response to deposition of immune complexes. And I've also been very interested in the function of these neutrophils in the context of autoimmunity. There's in particular autoimmune vasculitis, in which we found that they are activated in a manner that they produce copious amounts of these neutrophil extracellular traps, which then, for example, contribute to the kidney damage in these autoimmune vasculitis patients here. This is a glomerulonephritis section here where we see prominent deposition of these neutrophil extracellular traps. So I always, since I had this background in neutrophil biology from my PhD, they were always kind of on my map with regards to breast cancer. Now my lab studies breast cancer, and so I was always very interested in starting a project looking at the function of these neutrophils in tumor progression of the breast. And then luckily Hamad Al-Chitawi, who joined my lab, I think in 2017, if I'm not mistaken, was also very interested in these cells. And so we thought about a project together, and we wanted to go after their potential myeloid-derived suppressor cell function, which has been linked with these neutrophil granulocytes in the past. This is actually a collaboration with Craig Walsh at UCI, who was very supportive in helping Hamad and myself to think about this project, and also to set up some of the methodology to look at T-cell activation, etc. And so looking at these myeloid-derived suppressor cells, or short MDSCs, this is a heterogeneous class of myeloid cells that have been described in various contexts, including cancer, where they are able to suppress an adaptive immune response. They can do so through various mechanisms. They can directly suppress T-cell activation through various mechanisms that I'm not going to go too much into detail, but these include reactive oxygen species production, the function of arginase, but they can also indirectly interfere with T-cell activation through modulation of the antigen presentation machinery. So really, these MDSCs can sort of push on multiple buttons in order to suppress this anti-tumor immune response. So we wanted to study these guys in more detail in this project. And so first of all, we looked a little bit into the literature. There's good evidence that MDSCs are implicated in and have a clinical relevance in breast cancer. Without going into too much detail here, but there's numerous studies that show correlation of numbers of MDSCs in the primary tumor microenvironment with increased stages of breast cancer progression, as well as with metastasis. So there's a good evidence for the role of these cells in the breast cancer context in human disease. But in the next couple of slides, I just wanted to highlight a few, in my eyes, big gaps of knowledge when it comes to MDSCs in the cancer context. The first really being, where do MDSCs fit into the bigger picture of the cancer immunity cycle? So here, this is a nice illustration showing this so-called cancer immunity cycle, where number one, we have release of local tumor antigens, which have to be picked up by the antigen presentation machinery, such as dendritic cells, which then obviously migrate to the lymphatic system and present these tumor antigens to the immune cells, to the adaptive immune cells, such as the T cells, which then get activated and start to proliferate and migrate out towards the tumor itself, where they then can raise a tumor-specific immune response and start to try to eradicate the tumor. So where do MDSCs really fit into this larger context of cancer immunity? They could play a role in the lymphatic system, they could play a role in the circulatory system, or really in the primary tumor microenvironment or in the metastatic side. But I think one big question is really what is the bottleneck here in MDSC function? Which is something that ultimately we would like to get at. Another big gap in knowledge is really before we started the study, it was not fully understood how MDSCs really differ from their normal myeloid counterparts. So what I mean by this is that MDSCs have been shown to come in these two flavors. We have these monocytic MDSCs, marked by LY6C in the absence of LY6G, and granulocytic MDSCs, CD11B positive cells with high expression of LY6G. So we know that there are a subpopulation of these granulocytes and monocytes in breast cancer that can exhibit a tumor or immune suppressive capacity. But really, how do they differ from normal granulocytes or monocytes? That's been very elusive until recently. And so really what people have been doing mostly is isolate just this fairly heterogeneous population of CD11B GR1 positive cells, which contain both monocytes and neutrophils, and then subject these to, for example, T cell suppression assays and so on. But distinct markers and distinct expression signatures were not really defined for these granulocytic and monocytic MDSCs. So in order to study these MDSCs in the breast cancer context, we used a mouse model for breast cancer, which is the MMTV-T mouse model. It expresses this viral oncogene specifically in the breast epithelium that leads to constitutive activation of PI3 kinase pathway, phospho-AKT, and RAS pathway here. In a nutshell, leads to massive proliferation, particularly within the luminal compartment of the breast epithelium. And pretty quickly in these mice, you will get breast tumors that resemble a lot of the clinical characteristics of the human disease as well. So it's probably the most widely used spontaneous breast cancer mouse model. You do notice actually fairly early infiltration with leukocytes in this mouse model. And in previous studies, it has been shown that MDSCs actually arise in this mouse model, which made it a good model for us to try to go after the function of these MDSCs in breast cancer. So when Hamad started this project, he first tried to get a hang of this model a little bit. And we typically see palpable tumors form around 5 to 7 weeks of age of these mice, and then around 14 weeks they reach endpoint, and they also have metastases, particular to the lung. So what Hamad wanted to do first is to just monitor whether we see an expansion of these leukocytes here in various organs of these tumor-bearing animals. So he harvested them in late stage, and then he harvested bone marrow, spleens, lungs, brains, peripheral blood, and primary tumors. And without showing you all the data, what we found is there was a pretty significant expansion of CD11B GR1 cells in all of these organs. So there's a systems level sort of expansion of GR1 CD11B positive cells, which has been noticed before. But the question was, where are these cells most immunosuppressive? So can we find a very reliable source here of immunosuppressive myeloid cells? So for this, Hamad used this T cell suppression assay. So it's a T cell activation assay in vitro, where we're mimicking this CD28, CD3 co-stimulation without actually the dendritic cells being present. So there's only the antibodies that activate the T cells. And then you see profound T cell proliferation, interferon gamma production, which are the main readouts for T cell activation here in this assay. And then we exogenously add cells from these tumor-bearing mice or control mice where we know or where we suspect MDSCs to be present. And then we would, if MDSCs are present, we would see a strong inhibition of T cell proliferation, for example. So here's some of the data, and this is the data from the spleen, actually. So Hamad really compared systematically all these different organs where we saw an expansion of CD11B GR1 cells, but spleen really was the most predominant organ where we saw the most predominant suppression of T cell activation. And you can really focus on these two bars here, where we're looking at proliferated cells. When we add CD11B GR1 cells from wild-type animals from the spleen, there's no suppression obvious here, and comparing that to CD11B GR1 positive cells from tumor-bearing polyomyelitis T mice, both CD4 and CD8 T cells are strongly suppressed when cells are coming from these tumor-bearing animals from the spleen. So this gave us the rationale to actually isolate these cells now from the spleen and subject them to single-cell RNA sequencing so that we can try to reconstruct whether there's a subpopulation of suppressor-like neutrophils and monocytes in this fairly heterogeneous mixture of CD11B/GL1 positive cells. Quy Nguyen in the lab helped out Hamad here, being an expert in everything single cell. And the goal really here is to be able to reconstruct these subpopulation of cells, which would not be possible if we do a bulk level measurement. So just for those of you, I know it's now very widely used methodology, but just in a nutshell, how this works is it's a droplet-enabled microfluidics platform. This is a very slow motion of how these droplets are being generated, and think of these droplets as like individual reaction compartments that you can really create in a very scalable manner. And then cells are being distributed into these droplets, and they're paired up with these little beads that contain barcodes or barcode containing oligos, I should say. And these oligos have an oligo-DT component to them, so they efficiently kind of fish out all the mRNA based on their polyadenylation part, and then every mRNA that comes from one individual cell in one droplet will get a specific type of barcode. So what you can then do is you can break up these droplets and perform all this library preparation in bulk, but because of these cell-specific barcodes that are now introduced to every mRNA or every cDNA molecule, you can later on then demultiplex and generate these gene by cell expression matrices. So shout out to Nick Pervolarakis, who's a computational student in my lab, who was the main driving force behind the computational analysis for this first part of the talk here today. So just, you'll see a few of these dimensionality reduction plots throughout my talk. So what this means is every dot will be an individual cell, and the closer one cell is to one another, the more similar they are really in this highly dimensional expression space. So you can perform these clusters and then look at which genes are driving the differences between these clusters in a very unbiased fashion. So this is what the data looked like from our spleen derived cells. So we had wild type cells, and we had spleen cells from polyomyelitis T mice. So we essentially got 4 main clusters here of cells, and then you can look for specific gene expression to first identify which cell types are we dealing with here. So we found a large cluster of neutrophils here, marked by high expression of LY6G. There was a second largest cluster is the C1 cluster here, which are monocytes, marked by CCR2. But then there were also some additional cell types in there, a small population of T cells that kind of snuck their way into our flow cytometry here, and a relatively small cluster of B cells marked by CD19, for example. So we obviously focused on the neutrophils and on the monocytes here, and we wanted to know, are there particular subclusters here that are corresponding to the immunosuppressive cells. And one of the factors that had been described in the literature for MDSCs is IL-1 beta. They've been shown to express high levels of IL-1 beta. So lo and behold, within the neutrophils, we found this C2 cluster over here that showed particularly high expression of IL-1 beta. So this was, for us already, a good pointer that these could be the granulocytic MDSCs. Now, honing in on the monocytic cells over here, IL-1 beta was more diffusely expressed. So Nick decided to do a subset analysis of the monocytes, which sometimes allows you to get more distinct clustering. There we now see this emergence of a subcluster that is particularly high for IL-1 beta, potentially indicating our monocytic MDSCs. So now having these labels allows us to do all these comparisons that we previously thought are important questions to answer. For example, how do MDSCs differ from their normal myeloid counterparts? So this is what you can see here in this heat map. We have the granulocytic MDSCs and the monocytic MDSCs. They have a lot of overlap in their gene signature, in this immunosuppressive gene signature. But this is very different from normal neutrophils or normal monocytes, especially when you compare the granulocytic MDSCs to the normal neutrophil counterpart. There's really not too much overlap. Some of the classic genes are being downregulated, actually, including LY6G. Actually, we found to be downregulated in the monocytic or in the granulocytic MDSCs. And this is a little more digested here. You can see there's a fairly large overlap in the gene signature between the granulocytic and the monocytic MDSCs. And this gene signature involves, when we did GO-term analysis, to get a little bit more higher order information of what this gene signature may, what story it may be telling us. So it has to do with neutrophil activation. It has to do with cytokine-mediated signaling, etc., that promotes this MDSC-like response. And Hamad also very thoroughly confirmed some of these marker genes in additional mice by more targeted qPCR analysis. So from this, we're now able for the first time to really define how MDSCs differ from their normal myeloid counterparts. And we can also get some estimate of how similar or how different really these granulocytic and monocytic MDSCs are. And they do show a fairly large overlap in gene expression programs between these two flavors of MDSCs. So the next question that we wanted to answer is, how translatable is this MDSC signature really? Can we translate it to the human context?
So Kai, can I just interrupt you?
Oh, yeah.
Can you go back to the previous? Yeah, right there. So in looking at these, and it's a lot to take in at one shot.
Yeah.
What are you finding, or what can you point to that shows differences between the myeloid versus the granulocytic MDSCs in there? What you're looking at in panel I is all of them together, right? Because that's a GR1 positive.
Yes, yes. So, you know, here we're comparing polyomyl T versus wild type CD11B GR1 cells, assuming that, you know, in the polyomyl T mice, we get the subset of MDSCs. So overall, the expression level will be kind of pushed towards overexpression of these MDSC-related genes, even though we're measuring this in bulk here.
Yeah.
But we're able to pick this up because you know, it's expanding in the tumor-bearing mice compared to the wild type.
It's really interesting. I mean, the IL-1 beta you said is up here, but then when you showed the UMAT plots, you showed that the IL-1 beta was produced mostly by the neutrophil population, the granulocyte MDSCs.
Yes. Yeah.
So then going to the next one again. So you're saying then that again in panel I, when you see the elevated IL-1 beta, you can't really say from this that this is in the granulocytic MDSCs? Or you
yeah.
can't?
So I think really what's going on is I think the whole population shifts to some degree. So I don't think I have that data here, but you know, even so, we defined this cluster as our bona fide MDSC cluster, but you know, we have tumor-derived neutrophils within these remaining cells, which we sort of defined as sort of normal neutrophils, you know. But even within those normal neutrophils, you'll see some spots here of IL-1 beta, right? You can see that it's not there's no expression here, and it is.
The neutrophils express IL-1 beta, but it's interesting. It's only a subunit there.
Yeah, yeah, and in the normal cells, that is generally lower. I think there is generally a higher expression, even in the what we call non-MDSC neutrophils. So there's a push towards this whole signature that goes up. So we're trying to really wrap our head around what really is going on here. And I think actually we have some more recent data that may help kind of reframing this whole thing a little bit better, a little more systematically. But that's
before we go on. There's a note from Roberto saying, did you look at the tumor draining lymph nodes compared with the spleen? Are there any differences in frequencies or phenotypes?
Yeah, yeah. I think that's a good question. We haven't looked at the lymph nodes, but we did in a follow-up study that I will show some preliminary analysis in the last few slides of this talk. We did look at different compartments within the mice. We did not look on the lymph node level, but that might actually be a really interesting aspect, you know, because that's really where the main antigen presentation will take place, and you know, so that will be very interesting. That's a good point.
And then, just while we're interrupting you here, a note from Francesco asking, what about at the tumor mass level?
We'll get to that at the end of the talk.
Okay, thank you.
But those are good, really good points for sure. So back to this question, how translatable really this gene signature is. So we made use of this data set from Dana Pear and Alex Rudensky's lab, where they isolated CD45 positive cells from primary breast tumors and did single cell RNA sequencing. So Nick analyzed this data set, and we have neutrophils and monocytes to some degree covered here in this data set. And now comparing these cells with our MDSC gene score, so with our MDSC gene signature using this gene scoring analysis, you can see that those are the cells that light up. So the monocytes and the neutrophils really light up for our MDSC signature, which is not surprising. But then looking in more detail, in more granularity at the neutrophils, what was interesting is that we found, by unbiased neutrophil clustering, we found these 4 clusters of tumor-associated neutrophils. And cluster 0 in particular here showed a particularly very high similarity in their gene expression to our mouse-derived MDSCs. So this indicated to us that, at least to some degree, the signature is translatable into the human context. And you know, we now would really like to get our hands on some human tumor-derived samples as well, and try to validate this finding with, you know, more functional follow-ups. And one way that we could go about this is using CD84. So Hamad found that CD84 was very highly expressed in our MDSC cluster. So CD84 is a cell surface marker that has not previously been shown in the context of MDSCs. It's one of the SLAM family molecules, and very interestingly, actually, work from Edith Chacha's lab at the Weizmann Institute has shown that CD84, and it was in the context of chronic lymphocytic leukemia, can signal to surrounding stromal cells, which leads to overexpression of PD-L1 in the microenvironment. So this is something that could be very interesting. If these MDSCs are very rich in CD84, this kind of mechanism could explain some of the suppressive function mediated by MDSCs by, for example, leading to upregulation of PD-L1 in the tumor microenvironment, or in the spleen for that matter, or in the lymph nodes for that matter. So this is a very interesting aspect. So Hamad first went really thoroughly through validating CD84 as a marker for MDSCs in additional mice. So we see this shift of CD84 positivity, in particular in the spleen and in the tumor microenvironment compared to mammary fat pad. And this is quantified here. So CD84 does get upregulated pretty massively on these tumor-associated myeloid And then he also went through several functional follow-up analyses, which I'm not going to show all in detail, but it's kind of summarized here. We do see when he fractionated CD84 high and low cells, the CD84 high cells were really the ones that suppressed a T cell activation more robustly than the CD84 low cells. They also formed more ROS formation for more reactive oxygen species, and they showed high expression of our MDSC associated gene signature measured by qPCR. So we're very sure about this marker being a very good new marker for MDSCs. And that is actually the case as well for human MDSCs. We were not able to get cells from primary human cells from breast cancer patients yet, but this is something that we're working on. We're just setting up an IRB to collect blood from breast cancer patients. But Hamad was able to induce the MDSC program in vitro from blood PBMCs, and these cells do upregulate CD84 as well. So we do think that CD84 marks human MDSCs as well. So this would open up, obviously, to try to block CD84 using this inhibitory antibody from Edie Chacha's lab. And we're currently setting up an MTA with her lab to try to explore this idea, but we don't have any data yet on that. One particularly interesting aspect of this is how do MDSCs fit into the normal neutrophil maturation process. So neutrophils mature through various steps, and they some of you may know they have these specific granules, and each of these types of granules are being produced at different stages of neutrophil maturation until they really reach this final stage of neutrophil maturation, where you can also notice this polymorphic nucleus, and then they have all the azurophilic specific and gelatinase and secretory granules combined. So some of these genes can be used to reconstruct different maturation stages of the neutrophils. And so based on our single cell RNA sequencing data, obviously we have now single cell level transcriptome information for these cells. But the problem obviously is it's a snapshot in time when you do this kind of analysis. But you can assume that at any given time in your mixture of cells, let's say here in the spleen, we have mature cells and we have more progenitor-like cells present simultaneously at the same given time point. So one thing that you can do is the pseudotemporal analysis, in which you start to order all your single cell profiles and try to reconstruct these differentiation trajectories and how they may fit together. So we also used some additional single cell RNA-seq data from the bone marrow from Giladi et al. Here from Ido Amit's lab at the Weizmann, and they have looked at the whole bone marrow system and the whole hematopoietic system, including the granulocytic arm here and the granulocyte monocyte progenitors. So we had single cell profiles associated with the more progenitor-like stage as well, that we could use sort of as a roadmap for our own analysis. So this is what our data looked like. We pseudotemporally ordered these cells, and we found this sort of 3-branch trajectory. And this contains cells from wild type and tumor-bearing mice. And what you can see here, based on some of the genes, including MPO, neutrophil elastase, this marks more of the progenitor-like cells, actually. They're more of these early azurophilic granule-associated genes. So they're expressed in the neutrophil progenitor-like cells here. And then these move towards this branching point where we can see this clear sub-branch or branch forming here, which contains more mature neutrophils that are characterized by expression of several sort of antimicrobial genes such as CAMP and LTF. But then in contrast to those, we have this sort of aberrant form of neutrophil maturation that leads to our granulocytic MDSCs. So here we see IL-1 beta expression, and all the MDSC signature goes up in this branch here. So it really looks like this is a sort of a normally not occurring aberrant form of neutrophil maturation that gives rise to these granulocytic MDSCs. And here are some of the genes associated with the different stages. Like I mentioned, some of these antimicrobial genes are really upregulated in this arm here, but they're downregulated in the branching progression towards granulocytic MDSCs. In contrast, you get upregulation of CD84, IL-1 beta, and other factors. We were then particularly interested in this early branch here that got its own state, which we called GMDSC transition state. And this was associated with some interesting genes, including PIRB, which stands for paired immunoglobulin-like receptor B. And interestingly, work that was previously published has shown that this is an important factor that regulates the function and the fate of myeloid-derived suppressor cells. So that gives us more confidence actually that this transitional state may actually mean something. So it would be interesting for us to find specific genes that are being upregulated early in this process of MDSC maturation, which would then allow us to interfere with the maturation rather than dealing with the end effect of it when we have accumulation of all these cells. So possibly we could block the transition into MDSCs by interfering at this transitional stage. So this is something that we're following up on right now. Just to summarize what has been published so far, and there's please go ahead and check Ahmad's paper here. There's much more in the study that I didn't have time to present today. But in a nutshell, what we think is going on is that in these tumor-bearing animals, there's a vast production of GCSF and GM-CSF, which acts on the bone marrow. We get this sort of push onto granulopoiesis. We get an expansion of neutrophil progenitors. And we think actually that they have to go through multiple locations within the human body where they get educated towards this MDSC signature, towards this immunosuppressive capacity. One particularly important stage here seems to be the spleen, which was really the focus of this first study that I presented today here. And we think that this aberrant maturation process leads to, or we know that it leads to upregulation of certain chemokine receptors listed here. And this makes them more responsive to then get recruited to these active sites of inflammation, where they can then contribute to immune shielding and really be sort of a bad player in tumor progression. But obviously there are still open questions here, a lot of open questions actually, that we wanted to address, and that also some of the reviewers of this paper kept harping on. One being, so how do these MDSCs in the spleen now, how do they differ between the tumor microenvironment and the spleen? What happens in the peripheral blood, and how do they compare to the bone marrow, etc. And possibly I showed this slide earlier, but those neutrophils really are present systematically in or systems level wide in the whole body, including the lungs, which is a very prominent site of metastasis. So we wanted to see and get a more comprehensive view on.
Can I just interrupt you for a moment before we go on to the next part?
Yeah.
I mean, there is go back to your to the abstract, to the graphical abstract. Yes, this one. So, I mean, you think about the MDSCs, and it makes sense for the chemokine receptors. It makes sense for the genes associated with immunosuppression. But how does this, or how do you think this fits with the presence, or certainly the high expression of the neutrophil activation genes, and especially IL-1 beta, which very highly pro-inflammatory cytokine?
Yeah, that is true. IL-1 beta is very pro-inflammatory. I have to look back in this literature that implicated IL-1 beta directly in immunosuppression of the adaptive immune response, and I'm actually not 100% sure right now what the molecular mechanism of that was. But that's obviously a good point, because in general it would be pro-inflammatory. So that is a bit of a conundrum. But let me look up that paper. I can send it to you maybe afterwards.
Yeah, I mean, my lab's been very interested in neutrophils and production of IL-1 beta, and of course the whole processing of IL-1 beta in order to get it to mature.
Absolutely.
Let me just...
that could be a very interesting follow-up that we could think about addressing here as well.
Yeah, we should definitely talk afterwards. A couple of questions in here. One from Marianne.
Okay. For some reason I don't see the chat questions here, but please.
Well, I was about to read them to you.
Pass them on to me. Yeah.
Well, here goes. Unless, I don't think anything here is urgent. If you're close to finishing, why don't you do that? And then we'll just open it up for discussion.
Okay, sounds good.
Yeah.
So just to tell you what we've been up to follow up on some of these open questions, including metastasis. So lung is a very prominent site of metastasis in this breast cancer model, and we do see Hamad can see these accumulation of GR1 positive cells in these local foci of metastases in the lung. So to get like a really more comprehensive view on this, we decided to collect from every tumor-bearing mouse, we collect multiple organs to get like a really sort of whole organism view on this. And shout out to Alison, who helped us out with a lot of reagents here in order to set up our workflow and to optimize the tissue processing steps here, because this is actually not trivial to get all the different organs from the same animal and process them almost simultaneously to get them loaded onto the 10X at the exact same time. And what we decided to do here as well, which is pretty cool, is we wanted to look at 3 different animals, 3 tumor-bearing mice, and we wanted to keep that information for every individual cell from which mouse it originally came from. So we used this total-seq labeling, which is an antibody that contains a barcode, and then it's also being incorporated into your cDNA library because it has this this poly A tail here. So essentially, it gets incorporated into your data, and you can later on reconstruct whether it came from mouse 1, 2, or 3 here of these in this cohort of this for this experiment. So Yan Wen, who is a computational student in my lab, has been analyzing this data set so far. So this is very new. So we're still really crunching this right now. So I'm going to share just some updates here, and we're still trying to really get our handle of this data set at this point. But what we know is there's a large population of neutrophils here, multiple clusters within the neutrophil cluster, a smaller group of monocytes here, and we have fairly distinct gene expression signatures across all these different clusters. And this is actually representing the which cells came from which experimental mouse, and we have a very even distribution, so there's really no worries at all about something being an outlier in terms of coming only from one mouse at all. So that was very reassuring to us to have that information in there. Now, so obviously we collected these from all these different organs. We wanted to now know how different are they really. So within the neutrophil cluster, this is all tumor-bearing mice here. We don't have any wild-type mice in this data set yet. But just within the neutrophils, we can see that essentially we have this progression. So the bone marrow, spleen, and blood, not surprisingly, are more similar to one another, whereas the more peripheral organs really show a more distinct gene expression signature and makes them cluster a little bit separate from here. So these these lung samples are really mostly accumulating in this cluster over here. And the tumor-derived neutrophils are really accumulating in this cluster over here. Which is interesting because what we noticed is when we now look for some of these MDSC-derived marker genes, such as IL-1 beta and arginase expression, those were the highest really in these organs over here, in the metastatic lung and in the primary tumor microenvironment. And actually more on a systematic level, when we compared these gene signatures that we defined before, really it's the lung and the primary tumor in particular that shows really the highest level of MDSC signature, and even higher than the spleen and the peripheral blood in this context. So some of the things that we're envisioning doing with this data set is obviously comparing between the different sites, like how do these cells transition through the different organs. And you can almost think of this as like a pseudotemporal trajectory in my eyes, because we know the cells originate in the bone marrow. They will go through the spleen. There's some extramedullary hematopoiesis happening here, and some maturation that we know occurs in the spleen in tumor-bearing mice. And then they transit through the blood and arrive in the lung and in the tumor. So can we reconstruct this type of transition here? We're particularly interested in these transitional states between across the whole organism of the mouse to get a really more complete view on how MDSCs emerge in the tumor-bearing context. And then just another thing that we're very interested in is these ligand-receptor interactions. So what I just showed you is that in the tumor microenvironment, they seem to really tune up their MDSC capacity according to the gene signature. So is there something in the tumor microenvironment that promotes this? Are there factors that are produced by the local tumor cells that activate our MDSCs? So we performed this ligand receptor interaction analysis, and we found some first candidates here that are produced. So the tumor cell ligands are produced down here, and up here we see all the receptors that are expressed specifically on G and M MDSCs. So now we're starting to look into which of these could be potential candidates that may contribute to this maturation, this further maturation of the MDSC-like cells in the primary tumor microenvironment. So some ways to go about this would be to specifically overexpress some of these ligands in the polymodal T mice, then we can transplant these and determine, do we get more or less MDSC maturation in these overexpression. And an interesting candidate could be SPP1, which is osteopontin. That is one of the interactions here that has been identified using this analysis. So I think I already summarized all of this, all of what we think is going on. And we really want to try to translate some of these findings into possible therapeutics or even diagnostics. CD84, if it works well to detect MDSCs in the human context, could also be used in blood samples from breast cancer patients. And we could determine whether that correlates with poor disease outcome, for example. And then we're obviously interested in specific effector cell functions of how these MDSCs can directly interfere with T cell activation. I think this is it from me. So if there were any more questions, I'd be more than happy to answer them.
That's great. We'll start with the literal applause. Very good. That was really fascinating. Absolutely amazing, Kai.
Thanks, Eric.
And lots to talk about. There were 3 questions that came up. I'll start with Marianne. Marianne, do you just want to... Oh, wait a minute. We're going back. Yeah, so we talked about the tumor mass level that Francesco asked about. Armando asks, do MDSCs express PD-L1?
Yeah, they do. It doesn't always pop up in in our data set, but it's sparsely expressed and it's higher than in the normal neutrophils. So
Kai, my question comes from your point on CD84 when you had that nice diagram showing how you think that CD8, or at least one mechanism is that CD84 is acting through the stroma to turn on PD-L1.
Yeah. Let me go back. I just realized I totally botched the thank you slides, which Armando, do you mind if I quickly just say thank you to my team?
No, please go ahead.
Yeah, I totally botched this one. Thank you to the great atmosphere in the lab, and like lab meeting. That was, that's a lab outing here with Armando's lab and Devin's lab a little while ago. Now lab outings look like this, obviously, where everything is through Zoom. And this is actually from Hamad's birthday, and I wanted to really highlight Hamad's work again because he was really the driving force behind all this. And also, quick shout out to the U54 Cancer Systems Biology who supported this project with a pilot grant. And so I wanted to obviously shout out here to the U54. So with that, Armando, let me go back to your question.
Yeah.
I'll just jump back to the slides.
So as you're going there, Kai, what my question is, in your in vitro assay, if I remember the data correctly, you sorted out those CD84 positive MDSCs, but I'm assuming in that system, you don't have this third-party member, the stroma, right? But yet you still see a strong suppression. So does it suggest that this is actually acting more directly on the T cells, and it doesn't require this third-party mechanism that you're proposing on this slide?
That's a good point. Yeah, that's a really good catch. So that we don't know. We were trying to get these inhibitory antibodies to interfere with CD84 function, and like you said, we only have T cells and neutrophils and the myeloid cells together. So this would not involve any of the stroma here. And it could be that there's a direct role of CD84 in suppressing the T cells directly. Or it actually, that it could also be that the MDSCs in the whole body can prime the surrounding microenvironment to be more immunosuppressive, in addition to their direct T cell suppression capacity, which may act also through other mechanisms independent of CD84. So I don't know whether CD84 plays a role in this direct inhibition of the T cells.
So Kai, I'm sorry, Kai, related to that, do MDSCs make the same amount of free radicals that you see in neutrophils? I'm asking that question because free radicals suppress T cells.
Absolutely true. So that's what they do. They are producing a ton of ROS, actually. So that's what Hamad showed as well. And in fact, actually, speaking of that, we also looked into neutrophil extracellular trap formation, and what we think are the MDSCs are also more prone to net formation. So we actually already started working on whether nets themselves can inhibit and maybe scavenge the T cells, and suppress T cell proliferation. And then it turns out, just when we got some cool results there was an immunity paper that came out that actually showed that net formation is one of the mechanisms by which MDSCs suppress an immune response. So we got scooped on it, but it's also nice to see that we were on the right track there with this idea.
Cool.
But the ROS is probably one of the main factors in the T cell suppression.
Yeah, awesome. Thank you so much. Great work.
Thanks. That's a good question. Thanks, Armando.
That's interesting with the nets, and I'd like to see that paper as well. But just from that standpoint, it's the low density granulocytes that tend to be have a higher likelihood of forming nets.
Right.
That's been true in things like lupus and other autoimmune diseases, where they've actually identified, the Mariana Kaplan and others have identified low density granulocytes and shown that they are more active, and certainly in terms of nets. So Marianne, you had a question?
Yes, hi, Kai, that was really interesting. And
Marianne.
So thinking selfishly about work in my own lab, which is more focused on xenografting immunocompromised mice, you know, for PDX or other orthotopic injections. I'm just wondering if you know whether MDSC maturation proceeds as you have shown here, or whether it's different, stunted or skewed, or
In immunocompromised animals?
yeah, like in non-skid or NSG mice.
I actually don't know. This is something I would have to look into. Obviously, we don't have the immune system in those mice, which would be probably the main effect of these MDSCs. But you know, I think neutrophils in general are changing quite dramatically in tumor-bearing animals in general, and that could have other effects, you know, in the microenvironment. So I would not count them out, really, you know, in compromised. In fact,
I mean, I just think it's important to know, since most drug testing for FDA approval goes through these mice, and having a more sort of granular idea of how these immune cells differentiate and respond is very, very important. I think it's interesting.
Yeah, no, actually, that's a good point. You could test, for example, PDXs, you know, PDX models in patient-derived xenograft models, for those of you who don't know this acronym. So you get actually primary tumor samples from breast cancer patients or other cancer patients, and then you could still follow how do these types of tumors modulate this particular arm of the immune response.
Right.
Yeah, that's a good point. I don't know. I should look into that. Maybe Hamad knows something. I can ask him.
Or maybe you can just source some single cell data sets that are out there.
Right. Yeah. That's a good point. Yeah.
Francesco, you had a question that you may have answered already. I may have answered, but do you want to ask him?
Well, yeah, I had one initial question that is answered, and the one that I just wrote. So it looks like that your data are going into the direction of seeing or showing that granulocytic MDSCs are actually a lineage in themselves, and they are alternative to neutrophils, yet they arise from the same progenitor. Do you think you have enough data to say this is what is going on, or the opposite hypothesis, so that MDSCs and neutrophils can basically plastic interchange one and the other? Is that hypothesis still on the table?
Yeah, so I think the data that we have for this aberrant neutrophil maturation process is not very strong at this point. So I think there's a lot we could do to corroborate this further. You know, it's really just, right now, it's really just based on computational analysis of the data set. So I think there's a lot of room for further exploring this aspect. How do they really, how they really connected to the normal lineage of neutrophil maturation and the hematopoietic system in general. I think, you know, even though this is our model right now, but I think there's still a lot of room for further exploring this. You know, lineage tracing types of ideas we could explore. But I don't know. I mean, I haven't really thought about what would be the best methodology to follow up on that at this point.
Right. So then there's Martin. You had a follow-up question. Do you want to ask? Martin Mins. I'll just ask it. So along those lines, do you think these MDSCs can be reverted back to normal neutrophils, or do you think this is the end of the line for them?
I don't think they can be like reverted back or de-differentiated back into, I don't think that's, you know, like with neutrophils, they're short-lived in general. It seems to be more of like a one-way street, and then, you know, they undergo apoptosis. And considering that this MDSC state already seems to be more prone to net formation. That's a form of cell death. So I think they're pretty close to saying goodbye to this world, you know, and once they are in the MDSC state. So I don't think reverting. I think you know, since I have this up here, like interfering with this maturation process and somehow keeping it in check, and like keeping the maturation process, if this is really how it works, keeping it on the normal track, that, you know, with some pharmacological inhibition, that would be probably the way to go. But I don't think reverting, I don't think that would work. It's mine.
Right. Jim, you had a question?
Yeah, Kai, it looked to me like in the original cell sorter diagram that you showed, that you could have high CD11B without high GR1, but you couldn't have high GR1 without CD11B. Does that mean anything? Does it suggest that you have to have the one component before you can have the other?
Yeah. I think the CD11B high and GR1 low cells, they're probably more the monocytic and also the granulocytic monocyte progenitor cells. And then during neutrophil maturation, GR1 goes up, and I think that's how you get this spread.
Yeah, but you can't go the other way.
Yeah. At least, I mean, one thing that we notice is that LY6G actually goes down again in the MDSCs. So actually, it does seem to, the MDSCs are probably fairly low in this GR1 distribution.
Well, the LY6G is also can be an indicator of activation, and that it's involved in integrin signaling.
Right.
Itself, even though we all think of it as a marker for neutrophils.
Yes.
It does have a function.
Yeah. I think we're learning there were some other nice studies actually that I didn't show. There's a paper in bioRxiv on just general neutrophil heterogeneity, you know, in the blood, and it's actually they're more heterogeneous than we would have thought before. Yeah. So there's a lot of stuff going on with those cells that we don't really know yet what it means, I think, and so it's kind of puzzling with these kind of data sets, you know, so to try to make sense out of it.
Elise Greenberg, before she left, she was busy working on this single cell RNA-seq data to some extent with Qui.
Yeah.
I'll be in touch with you to talk about that.
Oh, that'd be exciting. That'd be cool. Yeah, no, for sure.
So, Will, you have a question?
I think it was already answered. So
okay, all right. Roberto.
Yeah, I had a question. Great presentation, Kai. Really enjoyed it. I noticed that in the images you showed from the spleen between wild type and tumor-bearing mice, that they seem larger.
Yes.
So I was wondering if you were able to quantify what that increase represented, because typically, you know, when we look at you know, viral infections or you know, autoimmune disease, we see this increase in the T cells or the B cells, and so you get these large lymph nodes. So I kept thinking, you know, if we have these suppressive cells, as you indicated, then I would have predicted, you know, less T cell proliferation, less B cell proliferation in the spleens from those tumors bearing mice. And so I was wondering if you knew the cell types.
Yeah, that's a good catch. I mean, we do see the splenomegaly quite dramatically, actually, in later stages of tumor progression. I'm showing the picture here. Can you see it?
Yes.
Yeah, we do know for sure that they have a ton of CD11B GR1 cells. So I think that population further expands in the spleens of these mice. I really don't know actually at this point. Personally, I don't know. I think the data probably exists. What happens to the other immune cell populations in there? But it's a good point. If you think, you know, there's a myeloid-derived suppressor cell that's massively expanding, why would you still see expansion of all these other immune cells? That's a good question. I mean, obviously, the MDSCs are just one button pushing on the whole immune system. you know, there could be a lot of other pro immune stimuli that are floating through these animals, you know.
Right.
So it's all kind of a matter of balance, right? Like if you take the MDSCs away, maybe you see even more T cell expansion in this.
Right, or maybe you won't necessarily see an increase in the number of the T or B cells, but perhaps that increase is from the myeloid compartment.
Yeah, it's that I'm not 100% sure. I know that the myeloid compartment definitely expands. Whether they account for most of this splenomegaly here, that I can't say for sure. But that's a good question.
I think at that note, it's 1:15, and I think we thank Kai again for a great presentation. Very, very evocative, for sure. And
thank you so much. Great questions too. I'll look up on that IL-1 beta.
Yeah, would you please? And I'll send you a note, Kai, and let's find the time to talk.
And that, yeah, we should definitely talk. And the net idea, I think I should send you that, because that's quite interesting.
I did not realize just how much of a neutrophil guru you are. I tip my hat to you.